Before talking about the mechanism of action of beta-blocking drugs, I'll just give a quick overview of adrenergic receptors, or adrenoceptors. Adrenoceptors mediate sympathetic activation. The body goes into fight or flight mode via activation of the sympathetic nervous system. Adrenoceptors are G-protein coupled receptors that are the targets of catecholamines such as adrenaline and noradrenaline. This binding leads to a physiological effect downstream, which typically results in an effect useful in fight or flight mode. For example, increasing heart rate to increase blood delivery to muscles and vital organs. Noradrenaline has a greater affinity for some types of adrenergic receptors, known as alpha receptors, and adrenaline has a greater affinity for beta receptors. This is why the distinction between alpha and beta receptors was made in the first place. Amongst these alpha and beta receptors are multiple subtypes, which are alpha 1, alpha 2, beta 1, beta 2, and beta 3. In this video, I will focus mainly on beta 1 and beta 2 receptors, as these have the greatest clinical relevance when explaining the mechanism of action of clinically relevant beta blockers. So the biggest pain when learning about adrenoceptors is learning the locations and the physiological effects of their activation. The two to remember for beta receptors, so if you remember anything, then remember that beta 1 receptors are found in the heart, and they lead to increased contractility and increased heart rate. And beta 2 receptors are found in the airways of the lungs, where their activation leads to dilatation. So first, let's start with beta 1 receptors. In the heart's muscle cells, also known as cardiac myocytes, beta 1 receptor activation leads to an increase in intracellular calcium levels, which increases the heart's contractility. So in fight or flight mode, the heart beats harder to increase cardiac output. Beta blocking drugs, such as atenolol, which is very cardioselective, prevent the binding of noradrenaline or adrenaline to the beta 1 receptor, leading to a decrease in intracellular calcium concentrations. This reduces the contractility of the cardiac muscle cells, leading to reduced heart contractility, also known as negative inotropy. The other type of cell found in the heart are the cardiac pacemaker cells, and these are found in the sinoatrial node and the atrioventricular node, and they are able to generate spontaneous action potentials themselves. They generate action potentials partly using calcium L-type channels. Beta 1 receptors found on these pacemaker cells, when activated by adrenaline or noradrenaline, increase the flow of calcium ions into these cells through the L-type channels. This enables quick generation of action potentials, which are conducted down into the ventricles, leading to a fast heart rate. Beta-blocking drugs block the binding of the catecholamine, therefore reducing the rate at which calcium can actually flow across the membrane and into the cell, and this means it takes longer to generate an action potential. At the sinoatrial node, this effect leads to reduced heart rate, also known as negative chronotropy. The same effect at the atrioventricular node leads to reduced conduction of the action potential, also known as negative gerometropy. The combination of these two effects is why beta blockers can be used as antiarrhythmics, for example in the treatment of AF. So that's an overview of how beta blockers act on the heart. Beta 1 receptors are also found in the juxtaglomerular apparatus in the kidney. So when blood volume is lost, this causes blood pressure to drop, the total circulating sodium drops, and this causes an increase in sympathetic activation. So we know that these three factors, so a drop in blood pressure, a drop in sodium, or an increase in sympathetic activation, induce the release of renin from the kidneys. And the release of renin causes an increase in arterial blood pressure via the renin-angiotensin-aldosterone system. So, beta 1 blockade also reduces blood pressure by way of reducing renin secretion. And this is why beta blockers may work in treating hypertension. Beta 2 receptors are found predominantly in areas of smooth muscle, for example, as previously mentioned, in the smooth muscle of the bronchioles in the lungs. Activation of these beta 2 receptors causes smooth muscle cell relaxation. So let's look at the lungs then. So when noradrenaline or adrenaline binds to the beta-2 receptor found in the smooth muscle cell of the bronchiole, this causes the smooth muscle cells to relax, leading to bronchodilation. If we give a beta-blocking drug, that's going to block the beta receptor, preventing the binding of noradrenaline or adrenaline, and then that will cause bronchoconstriction as the smooth muscle cells contract. So essentially, wherever beta-2 receptors are found in smooth muscle cells, they lead to smooth muscle cell relaxation. And another clinically relevant location to bear in mind is the smooth muscle cells of arteries, particularly arteries which supply visceral organs or the vital organs. So here, beta-2 activation leads to vasodilatation. And that makes sense because we want to improve blood flow. We want to increase our blood flow to our vital organs during sympathetic activation, during fight or flight mode. So essentially directs blood from the peripheries to the central organs. So if we give a beta blocker drug, that may actually cause vasoconstriction via beta-2 blockade. And this is why sometimes beta blockers can cause acutely a transient rise in blood pressure. However, this usually settles due to the reduced renin secretion that I talked about earlier. So they're the main clinically relevant effects of the beta-1 and beta-2 receptors and their blockade, basically. There are other locations of beta-2 receptors listed on this table, but they're not going to be covered in this video. I'll just note that there are currently no clinically relevant beta blockers that target beta-3 receptors, and so I've not touched on them at all in this video. There are many different types of beta-blocking agent. They each vary in their selectivity towards beta-1 or beta-2 receptors, and we divide them into three groups. The first group are the first generation beta blockers, also known as the non-selective beta blockers. These are drugs like propranolol, sotalol, and timolol eye drops, and they're deemed non-selective because they block beta-1 and beta-2 receptors. These drugs are more likely to cause side effects such as bronchoconstriction. The second generation beta blockers are known as the cardioselective drugs. This is because they target mainly beta-1 receptors, i.e. the drug has a higher affinity for the beta-1 receptor, which is found in the heart, hence cardioselective. This means these group of drugs are less likely to cause side effects associated with beta-2 receptor blockade, such as bronchoconstriction. However, it is important to note that no beta-blocking drug is 100% cardioselective, meaning that at high doses of cardioselective beta blockers, there will be some beta-2 receptor blockade, meaning that, for example, a high dose of bisoprolol can still induce bronchoconstriction. The third generation of beta blockers contains some cardioselective drugs and some non-selective drugs. For example, nebivolol is highly cardioselective, and labetalol and carvedilol are non-selective. However, they are put into a separate category because they have additional vasodilating effects. Remember, beta blockade shouldn't really cause vasodilatation because in fact beta-2 receptors, when blocked, cause vasoconstriction. However, these drugs work via different mechanisms. So for example, labetalol causes vasodilatation by inhibiting alpha-1 receptors, which normally cause vasoconstriction. Nebivolol works in a completely different way to adrenoceptors, and in fact increases the endothelial production of nitric oxide in order to cause vasodilatation. Some have suggested these drugs may be better for use in treating hypertension. So that's a very quick run through of the three main subclasses of beta blockers.